Thank you.
So today I'm going to talk about NOX4 and TGF-beta receptor signaling in regulation of anoikis resistance and metastasis. So my research is mostly focused on metastasis because metastasis is responsible for more than 90% of tumor-related deaths. So that's why we need to understand molecular mechanisms involved in metastasis to develop anti-cancer, anti-metastatic therapies. Actually, the metastasis is very complicated and multiple process. First, tumor cells, why it moves around, I don't know. The tumor cells detach from the primary tumor and invade into the surrounding matrix and enter into the blood vessels, that is intravasation. And in the blood stream, the tumor cells, they are circulating tumor cells, CTCs. The CTCs, they survive during the circulation, and they move out of the blood vessel so that we say extravasation. And these survived extravasated cells can form secondary tumor as a metastasis. So my research is focused on how CTCs survive during the circulation. Tumor cells in the bloodstream, they encounter very severe, harsh conditions such as shear stress, anoikis, which is cell detachment-induced cell death, and immune surveillance. And I will talk more about anoikis. So except blood cells, normal cells, they adhere to extracellular matrix, ECM, such as fibronectin, laminin, and collagen through the cell surface receptors, integrins. And these interactions give rise to survival signal so that cells survive. However, if cells lose their adhesion, then survival signals are blocked, but apoptosis signals are activated, leading to cell death known as anoikis. So anoikis is a detachment-induced cell death. Anoikis is very important physiologically because it prevents abnormal cell growth in the wrong place. So in normal cells, anoikis occurs naturally when they lose their adhesion. However, the problem is cancer cells. Cancer cells in the bloodstream, they are no longer attached to the matrix, and most of them die. That means anoikis occurs. However, some cancer cells, they develop resistance to the anoikis so that they survive. And the only survived one can go somewhere to make metastasis. So this data is our previously publicated paper. Here we have human normal lung epithelial cell line, BIS2B, and lung cancer cell line A549. And we cultured them in suspension, which is without adhesion. So normal cells, when they are in suspension, actually cell cluster together to make cell aggregate. However, in cancer cells, cell aggregation became larger and very quicker. If we count the cells, the viable cells, the cell number went down, which means that anoikis is sensitive. However, in cancer cells, with time, the cell number goes up, which means that they are anoikis resistant. Previously, we have also reported that cell aggregation formation is very important for the anoikis resistance. So if you have single cells in suspension, mostly they die. However, if you get some aggregation of the cells, they can survive from the anoikis. So the question is that how cells form cell aggregates. For this study, I used osteosarcoma, which is a very rare but very aggressive bone tumor. It usually happens children or young adults with lung metastasis at diagnosis. So osteosarcoma is a good model for the study for metastasis. So this, oh my God, it is. So the importance of the ECM changes in osteosarcoma progression and metastasis is reviewed in this review paper. And they usually focused on how ECM changes are involved in cell adhesion, invasion, migration, metastasis. But my research is focused on anoikis regulation. So here we cultured the normal bone cell and osteosarcoma cell lines without attachment in PBS, media without serum or with serum. And we stained the cells for live dead assay. So live cells appear green, red, the dead cells appear red. So if you see more red cells, that means that anoikis occurs. Here, in PBS, cell aggregation is very low, but in the media, cell aggregation gets bigger and further increased with the FBS. And the red cell, the dead cells, some populations are red, but in the media or with the serum, you can see less red cells, which means that in the media or with the FBS, the anoikis is blocked. These data indicate that cancer cells produce protective factors to overcome anoikis. So we performed RNA-seq analysis comparing attachment and suspension. So with this analysis, we found that genes related to extracellular matrix, cell migration, are very upregulated in suspended condition. In terms of biological process, extracellular matrix organization and structure organization and cell matrix interact adhesion, migration signalings, those pathways are upregulated upon cell detachment. So we looked extracellular components and we compare suspension and attach. We can see, this is very interesting that we can see compared with the attachment in the suspension, fibronectin protein levels went up in the U2S and 147B, which are osteosarcoma cells. However, there is a little difference between attachment and suspension in normal cells. So indicating that cancer cells somehow they produce their own ECMs upon detachment. So here you can see fibronectin upregulation with the time, although the time kinetics are different. So the next question is that then fibronectin is secreted to the media. So we collected media from attached cells and suspended cells. And then you can see here, In suspended cells, more fibronectin is secreted. So we knock down fibronectin and we suspended the cells, and then you can see here more apoptosis occurs. And if you add fibronectin, then cell cluster became bigger and less anoikis we can observe. To test this in vivo, we injected protein 147B cells that was knocked down, the fibronectin was knocked down. We injected cells via tail vein, and you can see that a fibronectin knockdown group, there is a little lung metastasis with lighter lung mass. And the fibronectin knockdown was observed with IHC. So the next question is that how cells upregulate fibronectin when cells are detached. Previously, we have shown that with these two are our lab. We reported one. So TGF-beta receptor activation is very important for the osteosarcoma proliferation and EMT-like phenomena and the cell invasion and migration. And also this paper is very classic paper done by Joanne Massek, demonstrating that TGF-beta stimulation increase expression of fibronectin and collagen. So we examined cytokine expression in the osteosarcoma data set, and we found that TGF-beta 1 in all the osteosarcoma cell lines are significantly upregulated compared with other cell types. So we looked whether TGF-beta is secreted upon cell detachment. And we can see that compared with attachment in the suspended cells, the cells secrete more TGF-beta. And from our RNA-seq data, we also found that TGF-beta receptor signaling pathways are upregulated. How about TGF-beta receptor? Very interestingly, also we can see that TGF-beta receptor protein levels were upregulated upon cell suspension, and also its signaling pathway is activated, determined by phosphorylation of SMAD2. And what happened if we knock down TGF-beta receptor 1? And as you expected, you can see more anoikis happen here. And if you knock down TGF-beta receptor 1, then phosphorylation of SMAD2 is inhibited together with fibronectin decrease. So if we treat with the TGF-beta receptor inhibitor, then you can see cell cluster formation is decreased together with the fibronectin reduction. NOX4 is the one that we previously reported that it is increased upon cell detachment and for fibronectin secretion. And here in osteosarcoma cell lines, you can see more NOX4 expression, and ROS formation is increased. And if we knock down NOX4, then anoikis is decreased with downregulation of fibronectin and TGF-beta receptor 1. Epocyanin is a NOX4 inhibitor, and also it induces anoikis and with the fibronectin decrease. If you overexpress NOX4, then this is very important one, that fibronectin and TGF-beta receptor is upregulated. And we saw this, if we knock down NOX4 and then injected cells via tail vein, then you can see less metastasis with the decreased TGF-beta receptor and fibronectin levels. So what is clinical correlation of this? And we have osteosarcoma patient tissues, and we stand for fibronectin, TGF-beta receptor 1, and NOX4. And then most of the tissues are positive for these three proteins, and the correlation, co-expression was positively correlated. And this is not significantly, there is no significance because of the sample is not big enough. So we did again samples from Seoul National University, and then we found that all the samples, most of the samples are positive for these three molecules, and NOX4, fibronectin, and TGF-beta receptor NOX4 co-expression was highly correlated. With the public data, we also saw that compared with the normal bone cells, tumor cells have more fibronectin, TGF-beta receptor, and NOX4, and also their co-expression was very well correlated. So as I told you before, the osteosarcoma is a very rare cancer, and we don't have enough survival data, but we can see the tendency that the patient with higher fibronectin, TGF-beta, and NOX4, then survival with metastasis is very poor. So with this data, we proposed this model. In the bloodstream, if the cells make their own matrix quicker enough, then they can get some cluster, cell cluster, and then they survive. For the mechanism, we propose that cell detachment induce TGF-beta secretion, and NOX4 stabilize TGF-beta receptor, so the receptor is upregulated, and these two actually activate TGF-beta receptor signaling to make fibronectin upregulation and secretion, and through this integrin interaction, then the cells can survive. I have two more slides, that is. So currently we established this orthotopic osteosarcoma metastasis mouse model, and with the metastasized mice, we took the tissues and we make the cell lines, and also we took the blood, and then we are established these CTC cell lines, and then we will compare how they are different in terms of metastasis regulation. Lastly, this is one of my favorite slides, and then I would like to thank the people involved in this study. And most of the data shown today is done by Gan Lim. Some of them are Ji Young Song, and Dr. Kim and Dr. Park and Eugene Huang are recently joined members. And I thank to Center for Rare Cancer for their providing samples and discussion, and the pathologist. And we have a research core center for the animal facility and facts analysis and confocal microscope. And we also have the collaborator outside of our center, and we also include this NICOM. Okay, thank you very much, and I'm happy to take any question.